Title of our talk today is Human Subject Research Protocols. If you write it well, they will come. Bonus points to anyone who got the Field of Dreams reference. But with that, we'll go ahead and get started. So just a quick overview of our kind of presentation today. We'll have a brief introduction of myself, kind of a run through the standard DMCC protocol review process. I'm aware most of you probably have been through this, but just kind of a refresher on that process. We'll have kind of a touch base on some of the DMCC resources that we've put together to help you in creating your protocols. And then we'll try to highlight section by section the different protocol sections that should be in your protocols that will help assist in creating a effective protocol. And then we should have some time left over at the end for some questions. So I would ask that everyone kind of hold your questions until the end, and we'll try to get through as many of them as possible, if there are any. Just a brief introduction about myself. My name is Mike Fusakio. I have a PhD in biochemistry and molecular biology from Indiana University. I have experience in both the contract research organization and academic regulatory landscapes. I spent 2 years as a medical writer and regulatory affairs project manager at MedPace, responsible for creating protocols and clinical study reports, as well as being the regulatory lead for the successful submission of multiple INDs and an NDA to the FDA. While at Cincinnati Children's Hospital, I've spent about 1 year now as a senior specialist regulatory affairs, working on generating new content for the DMCC and for reviewing protocols for the different consortia, as well as working as a regulatory lead for IND submissions for the groups here at Cincinnati Children's. Now this next slide is one that you probably have seen before. This is kind of just a general overview of the DMCC protocol review process. What ends up happening is when a consortium sends a protocol to the DMCC, we'll take an initial review by your regulatory project managers. They will then pass that along to the group that I work with, where we will do our regulatory review. We will then pass it along to the data management group, and the PIs of the DMCC will do a review as well. Several of you have been on calls that we've had to kind of discuss the protocols at that point, where we kind of pass along our suggestions on things that we consider altering or changing that could help improve the quality of the protocol. You should be able to take the protocol synopsis, throw out the rest of the protocol, and still have a very good idea of what you're trying to accomplish, who is going to be in the trial, and what analysis will be done with that trial. So the text in the synopsis is always suggested to be the same to the text in the body. The reason we kind of give this suggestion is that when updates are necessary for your protocol, you know that the text is the same in the synopsis as in the body of the text, and it's very easy then to replace text throughout the protocol. So you can see here on the left, we just have some general sections that are kind of major topics for your protocol, that being title, study objectives, study design, inclusion, exclusion criteria, and statistical analysis. So again, these are kind of more of a summary, but it should reflect what's present in the body of your protocol. One of the sections that we highlight in the grant to protocol conversion document is the protocol background. The reason we highlight it is because it's very different than the specific aims, significance, and innovation sections of your grants. A protocol's background section should succinctly introduce the disease, discuss any previous non-clinical or clinical work, and then if your study involves a drug or a device, introduce that drug or device along with the proposed risks and benefits and an analysis of the risk-benefit ratio to that intervention. One thing the DMCC has kind of requested in this section is that a subsection be added to discuss how this protocol will lead to clinical trial readiness. Since this is a major objective of the RDCN, which your protocol should try to align to. The specific aim section of your grant will end up being better used by the study objective section of your protocol, as that will be more aligned with aims or the specific goals of that study. So we want to highlight that section here. The study objectives should be clear, concise statements that are being evaluated during the study. Your grant hypothesis can be the basis for which you use to create your study objectives, but we recommend against generally just a copy and pasting of your grant hypotheses. You can separate your objectives into subcategories. Those are generally primary, secondary, and exploratory. The presence of these subcategories is often dependent upon the type of protocol that you are putting together. It might be dependent upon the phase of study you're looking at and whether or not there's a study intervention or as a drug or a device. This is obviously something that you should consider when putting together the study initially, and often something that the DMCC would be happy to review. The study endpoints, which should detail the specific measurements then or outputs that will support the evaluation of your study objectives. So each study endpoint should be associated with a study objective, and since they're associated, these endpoints then can be divided into those same primary, secondary, and even possibly exploratory categories. We do see occasionally rationale for objectives and endpoints, but you don't need to provide that information. The rationale often can be covered based on the background information that you provide. It should be relatively clear why then you're evaluating these objectives and these endpoints. So the specific rationale does not need to be provided in the study objective or the study endpoints sections. One thing that I always recommend is that you should compare your study endpoints with your schedule of activities or schedule of events table to make sure that you've accounted for every endpoint and output that you need to measure during the study in your particular study visits. Your study design section, which will be after objectives in the DMCC template, starts with a study schema subsection. The study schema is a really helpful way to illustrate the overall design of a protocol. It's a good entry point for new study team members, as it's an easy way to kind of understand the overall flow and arrangement of the study. These become incredibly useful when there are multiple visits or multiple arms to the study. One thing we recommend is that any figure legend describing a study schema provide details and the definitions of any abbreviations that you use. What I've provided here below is an example from a clinical trial protocol from LIB Therapeutics that I pulled from clinicaltrials.gov. The nice thing about this study schema is it's very easy to understand how the study was performed. You can clearly see there's a double-blind treatment period, there's a 4-week washout period, and then an optional longer-term extension period that lasts for 12 weeks. You can easily tell that there were 4 arms to this study, 3 of which involved the study drug, and 1 in which involved the placebo. And then you can see on the bottom that they clearly provide abbreviations and their definitions that are used in this figure to allow for easy understanding of the study schema. One of the things that often kind of gets missed in clinical or human subject research protocols is discussion of enrollment and retention. All protocols should discuss how recruitment and retention will be handled. Study retention becomes very important when your study involves a high number of visits, any type of invasive procedure, or a long duration. You're asking a lot from your subjects, and even though they're often very motivated to participate in the trial, you're still asking for a large time commitment from them. So you need to consider how you're going to be able to keep them in your studies. You should also consider how screen failures will be handled. Will your protocol allow them to be rescreened? If they are going to be rescreened, is there a necessary time that subject must wait between their first screen fail and the time in which they are attempting the screening again? Is there a limit to how many times they can be rescreened? And then one of the things to always consider just from a cost perspective is if you are going to allow rescreening, does that subject need to repeat all the screening tests again, or are they able to only do a small subset based on the results from the first screening? One of the other major things that you'll want to consider in the study design section is the subject withdrawal. This can be subject withdrawal due to the patient withdrawing consent or being removed by the PI of the study. So you want to determine the circumstances that will lead to this. That would include withdrawal criteria and just the general patient withdrawing consent. You need to consider if a subject withdraws, are there special procedures that need to be followed? Now, this is more important generally in studies that involve some type of study intervention, where often there's a concern about a sudden withdrawal from a medication or the need to remove the device, but it's still something to be worth considering, even for if there's no study intervention, if there's any special procedures that need to be followed. An early termination visit is often a very useful way to try to get any last minute data from a patient that is going to be removed from the study. And if you're going to be including an early termination visit, it should be explained on your schedule of events so that it's clear what procedures are going to be performed on an early termination visit. You need to consider whether that subject will be replaced. If you're attempting to achieve its target study size, will you replace that subject or will it just be not replaced? Again, as I noted earlier, this criteria for study withdrawal are often more important for studies that involve some type of drug or device, but especially for even non-intervention studies that last a long period of time, study withdrawal is a major concern and should definitely be considered when writing your protocol. I think one of the two major sections of the protocol that are kind of the most important, in my opinion, are the inclusion-exclusion criteria and your procedure section. And so we really want to spend some time kind of on the inclusion-exclusion criteria to kind of help kind of better craft these sections. Obviously, well-designed inclusion-exclusion criteria can help avoid unnecessary protocol deviations and potentially missing out on potential subjects and help potentially speed up recruitment. So you need to consider what activities are going to occur during screening. Discuss which inclusion-exclusion criteria can be completely evaluated during that first screening visit. So you need to consider what can be found in the medical records versus what needs to be collected on site on that day. Do any of those on-site tests require being sent out to an outside lab? And if it's sent to an outside lab, how long does that take to get those results back? And so if you're requiring some kind of result that's going to be take a day or two or a week, then there's the understanding that you're going to need to have another visit potentially to review those results before they could potentially move forward in the study. We always recommend that you review your inclusion-exclusion criteria against each other. This will help avoid any kind of mirroring or redundancy. I've seen it in protocols both on industry side and research side, where the inclusion criteria notes patients between the age 8 and 18, and then their first exclusion criteria is patients under 8 and over 18 years of age. You know, they're both kind of targeting two separate groupings, but they clearly state the same thing, that the patients have to be that age 8 to 18. So you just want to make sure that those type of things aren't necessarily included, because it's just one more thing that your team has to kind of check, and it's one more thing that you can kind of remove to speed up the process during screening. You'll definitely always want to consider the time frame of your study. Will a subject fail an inclusion-exclusion criterion at a later time point based on how your criteria are written? For example, we have here patients greater than or equal to 8 and less than or equal to 18 years of age. Well, let's say this study lasts for 2 years and patient number 7 enrolls at the age of 18. When they enroll, they are within that criteria. But if the study lasts 2 years, then by the time the study ends, they would technically be outside of that criteria. So a good way to fix this is just to consider adding the phrase at the time of screening to that criteria, and then you don't have any concern about the duration of the study. One thing I always recommend to PIs is the use of a catch-all exclusion criteria. It's a useful way to ensure that subjects that may have any type of confounding conditions can be excluded from the study. And we've provided an example text here that you can use if you'd like to in any of your protocols. And it just has any other finding which in the opinion of the investigator would compromise the subject's safety or participation in the study. This allows the PI or the investigator to basically review, when they're reviewing potential subjects, if there's some concern for the safety aspect, basically exclude that patient. As always, if your study is going to involve children, which many of yours do, remember that you're going to need both informed consent and assent. And that also means you need to consider whether subjects, when they reach the age of adulthood, will be needing to be reconsented. And one last little formatting kind of tip or trick that we always suggest is to number your exclusion and inclusion criteria. And the reason we suggest this is that if you're having problems with recruiting, evaluating your screen failures with numbered inclusion-exclusion criteria allows you to see which criteria are causing the most problems. And if a change needs to be made, you're able to better evaluate that need and make the change to hopefully increase your recruitment. Another section that isn't noted that I think is really important is your study procedures section. Your study visits, even if you have just one study visit, should be appropriately detailed. We kind of always joke, but you should basically assume maybe that your study coordinators may only review this section. I'm sure the background's going to be great and really interesting, but they may just probably only review the study procedure section. So you need to consider what occurs, and more importantly, do those events that occur have a particular order that they have to occur in. One of the things that we see a lot is that there's no specific visit windows. So you always want to make sure that you specify visit windows for the study visits, but that those study windows are reasonable. As you all know, a lot of your studies are multi-site and you're pulling in patients from all over the world or even the country. And so you have to kind of remember that a one-day window might not be reasonable if a subject has to fly in from across the country. This schedule of events or schedule of activities or procedures, however it's called in your protocol, is an incredibly useful tool to ensure consistency at your site, to ensure consistency across all the sites and throughout the protocol. The SOE, as I'll call it, should be present, have each visit window, each study procedure, and the information needed to perform those procedures. So if there's particular timing, and if you're going to use abbreviations in your schedule of events, which we would recommend because it saves space, you should definitely provide abbreviations for those definitions for those abbreviations. As we noted previously, make sure that you always compare the schedule of events with your study endpoints to make sure you're kind of hitting all the necessary endpoints that you said you were going to evaluate in the study. There are kind of your standard study procedures that you should always be included. Those include adverse events, concomitant medications, informed consent. Those items are always important to include, even though they're kind of standard and they happen in almost all studies. We should always include them in the SOE just to try to make sure that there's consistency and something isn't missed. Another small recommendation in terms of formatting is that if your schedule of events ends up being more than one page, to repeat the header row, because there's nothing more annoying than scrolling down to the second page of the schedule of events and then having to kind of try to pair up the two pages to make sure that you're still looking at the same day. Like most of the things in the protocol, the schedule of events is a little bit of a balancing act. You want to provide a detailed explanation of how what's occurring, but the specifics on how a particular test is performed or how a particular procedure is formed will be better suited for your manual of procedures or manual of operations. The reason being is that this wording is often this information is often very wordy, or it will change frequently, whether that's due to software versions, hardware models, or kit numbers. And those kind of changes then require amendments that aren't really important. What I've pulled here is an example of a fairly well done schedule of procedures. This is also from that same LIB Therapeutics clinical trial from clinicaltrials.gov. As you can see, along the header row, they clearly state the study date, the visit number, and provide the window for that visit. And in the far right column, you can even see that they have an early termination visit and the procedures that occur on an early termination visit being the same as their final visit. On the left, you can see they list all their necessary study procedures. So you have kind of the specific ones to this study, which are like the PCSK9 measurement, but your more standard ones as well, like adverse events, randomization, informed consent, inclusion and exclusion criteria. I didn't include it, but on the continuation of this schedule of procedures in the protocol, they do provide the footnotes and the definitions for all the abbreviations that are used in this schedule. One thing we always recommend is that you kind of review the procedures that you'll be using in this study with any participating sites if your study is going to be multi-site. If you plan to use standard of care procedures, you want to make sure that's consistent across your sites for the reason that it's best to have all data points collected, or you may not be able to compare all possible endpoints if a test is not performed at one site. Just consider the fact that even something as simple as a physical exam may be different at different sites. So that means not only will you want to kind of properly describe what a physical exam is, but you want to make sure that it's being performed consistently across your sites. Always kind of consider the time it takes to perform each procedure and whether those necessary tests can occur within one study visit. Also be careful when you do specify time because it can cause some issues in terms of protocol deviations. The example we've provided here is the patient will sit for 5 minutes before blood pressure is taken. This is very standard text that we see in a lot of protocols, both industry side and academic side. The problem with it is it means that the patient has to sit for exactly 5 minutes, or it is technically a protocol deviation. So what we would recommend is changing this text to the patient will sit for approximately 5 minutes before the blood pressure is taken. This is basically the same text, but it gets you out of a potential protocol deviation. I am not a statistician, so I can't give you a lot of information on your statistical analysis section. However, we always recommend that you involve your statistician early in the process. If they can be involved in generating the hypotheses, the design of the overall study, that's great. If that isn't possible, they should at least potentially have a statistician review your protocol and review the protocol section on statistical analysis. And just to make sure that you always can have your statistical analysis section considering both the objective sections and your endpoints and whether the feasibility of achieving those two sections with your planned study size. And then obviously, if necessary, to do any type of power calculations to evaluate the actual study size of your potential study protocol. These are some smaller sections that are present in your protocol that we didn't think necessarily warranted a full slide, but definitely want to kind of touch base on because they are kind of important sections as well. Make sure you're always including a risk-benefit analysis for the overall study. So you want to provide information on what are the risks associated with being in this study, if there is any potential benefit with this study, and be honest in this section. If there's no benefit for the subject, just be honest that there's no benefit outside the gain gaining of knowledge. If your study will involve an intervention, study drug, we'll need to highlight the study drug supply, the preparation and handling, who's going to be taking care of that, how it's going to be done, administration by the subject, or will there be personnel administering the drug, what speed, if it's an IV drug administered, how the drug will be distributed to the subjects, how in terms of compliance and in terms of taking the drug. Again, this is a protocol, so broad descriptions with more detail being provided in your manual of procedures. If necessary, you'll want to have sections on how subjects will be randomized, what are your blinding and unblinding procedures if you're going to have a study drug or device and you need to address those concepts. We have some template text in our document on data management and monitoring plans, but you need to consider if those are enough for what your study requires. Also providing some text basically on publication plans, so that's clear as well. The really important always sections as well of human subject protection. So you'll want to cover privacy and confidentiality. Most of your studies end up being for the RDCRN, sponsored by the NIH. So there should be a discussion of the certificate of confidentiality. You'll want to discuss adverse events and how those will be reported. You'll want to discuss the informed consent process and assent process, if applicable, based on your study population age, and then the funding source. And then whether or not you'll be compensating the subjects. A lot of times we see additional documents provided with your protocols. And these can kind of be recruitment materials, newsletters, questionnaires. A lot of times the questionnaires are the ones we see most commonly provided as appendices. These are important and they'll need to be reviewed from an IRB's perspective because they will be often subject facing. However, they aren't necessarily needed as an appendices to the protocol, and we often kind of recommend that they are, just for the sake of the fact that these kind of documents tend to be updated on a much more frequent basis, which would require more amendments for your actual protocol. We'll kind of finish up here with just some additional tips and tricks on protocol that we have kind of put together that kind of hopefully help you in terms of putting these together. I completely understand, but you know your terminology within your field, and you know your abbreviations, and it's real easy to get in the habit of just throwing them out and kind of forgetting that to anyone outside your field, that's not just like the most common thing ever to say. But always remember that new team members or an IRB or potentially an FDA reviewer may not know your terminology or your abbreviations. So what we recommend is that you introduce your abbreviations at first use. Just use the abbreviation and the definition. And our DMCC protocol template has an abbreviation list. This is an easy and helpful item to add to any protocol. We include ours right after the protocol synopsis, just lists every abbreviation that's used in the protocol and has the definition written right to the right of it, makes it easy for people to review. Always be cautious about specifying that a particular individual must perform a particular task unless there's an obvious reason that that's required. So you can always kind of be general in that sense of saying a study team member, the radiologist. You want to be careful because otherwise you'll lock yourself into having to have Dr. So-and-so do this task or be unable to do the task because Dr. So-and-so is not available at that time. We are completely aware that these kind of multi-site protocols or even single-site protocols are often the collaboration of several investigators, and so it's very common then for them to kind of be piecemeal together. So what we always recommend is when you kind of getting close to completed, just a general review of your document, look at it for the sake of font size, your font styles, the margins of the page. A lot of times that can be issues with, your schedule of events is too wide and it can cause an issue if in case the document's printed off, you might lose some of that content. One of the things we do really recommend is that your figures and tables are looked at to be appropriately sized. One thing that's common is that the text is often wrapped around a figure. This is, I think, again, something where going from a grant to a protocol. In a grant, this is absolutely what you should do because there's the page limit concern. But in protocols, there's no page limit. And so we recommend against text wrapping just because it makes the text a little harder to read. The figure tends to be smaller, and it can really cause some issues later on if it's transferred into a site-specific document or if any future amendments are made to the protocol and things are shifted around. And kind of the last one that we have really is once you're done, just review your references that you actually used within the content of the protocol and just make sure that you're only citing references that were actually used. A lot of the citation managers kind of take care of this, but if you're not using one or haven't updated it, just make sure that you kind of real quick check that before sending it off for review.